students, welcome to lecture 2 of week 5. So, in this lecture we will look at what are reads, and in the next lectures basically we will be dealing with the assembly and different types of assembly methods for different types of sequence data. But before that, the reads that we generate from multiple type of platforms, technologies, right, they should be cleaned up before you take them for the assembly. Otherwise, cleaning is of also various types, which I will discuss. Otherwise, basically if I don't clean the reads beforehand, the raw reads that we generate from sequencer, right, has a lot of other sequences, unintended sequences such as adapters, primers, okay, which are used during the library preparation method, which help in the sequencing method, and they are like specific to different methodologies. But we have to remove these sequences, otherwise they will interfere with the process of assembly and also your downstream analysis, because you do not want these sequences as they are not part of your genome. Okay, so before going for a genome assembly, we should first understand that we will just see a quick overview of short and long repeats and the methods that we use to generate them. We have already covered it, but just a quick overview, and then we will look at how we are processing these reads so that we can clean these reads, and also for the genome analysis, in addition to the assembly, you also need to determine like what is the ploidy level of the genome. It's a diploid, haploid, triploid, okay, and then basically also the heterozygosity, because less than 1% heterozygosity genomes may be easier to assemble, but when the heterozygosity increases, they pose several challenges for the assembly, right. So these kind of things we should know beforehand, so that before going for the assembly, and they also help in estimating that how much sequencing may be needed, as we have discussed in the previous class. And also like at this stage you can also decide like if you want to go for more sequencing or not. Okay, so so far we have seen there are two types of reads that we generally use: short reads and long reads. Short reads are generally produced by Illumina or ion torrent sequencers, which produce reads up to 75 base pair to 300 base pair, but generally we go for 150 base pair reads, which could be paired end also. You will see the paired end thing. And similarly, long reads can be generated by ion torrent or not ion torrent, ONT technology, that is the Nanopore, ONT Nanopore, or the PacBio sequencer, right. So basically these can produce an average length of 10 kb, which is huge, and also it can go up to like several hundred kbs in length, which helps in the assembly method. So basically a combination of short and long reads help in the hybrid assembly approach, which is important for achieving higher contiguity as well as the accuracy. So short reads generally by sequencing by synthesis method is the most common, but sequencing by ligation is also available, and basically Illumina is example of sequencing by synthesis, which produces short reads, very high throughput, low cost, okay, highly accurate. And ion torrent is another technology which can also be used, but basically I think the market leader is still like Illumina in terms of reads for genomics, right. So, and the long reads are produced by ONT or Nanopore or the PacBio, of which PacBio is little expensive in terms of instrument and also the running cost, whereas ONT is more affordable, but PacBio is more accurate compared to ONT, though ONT is also increasing. Nanopore is also increasing in terms of accuracy, and both can produce long reads of like several tens to several hundred kbs in length, okay, which are very good for the assembly, right. And both have different methodologies, and that is why they will have different types of adapters and trimming that may be needed. Okay, so basically this is a overall summary that we will be using short reads and long reads, and again link reads also like 10x Genomics or TEL-seq by the Illumina, so which is basically giving information about fragments which are like sequenced like separately, because they have different adapters attached to the partial subsequences that we derive from those fragments, and they help in like long distance assembly or basically improving the assembly. And the sequencing is generally done by after the libraries preparation is done at by 10x, the sequencing is done by Illumina. Okay, so PacBio Hi-Fi can produce up to 99.9% accuracy, which matches the Illumina at this point, but Nanopore may be little expensive in terms of the errors, right? And both together can help you to achieve chromosomal level assemblies or even telomere assembly that we have seen in the case of human genome. Okay, so this tells like there are 3 different technologies: PacBio, ONT and Illumina, which produces different types of lens, and different type of platforms are available, like in case of RS2 sequel 2, and in case of Nanopore, MinION, GridION, Prometheon. So basically these are like, if you see all these like different platforms will have same chemistry for a particular company, but they have differing in terms of their output or cost, because if it has more output then the cost will become lower. But sometimes you do not want a very high throughput machine, because that may be very expensive for your research purpose, and you would go for a medium or lower level machine. Okay. And like similarly for NextSeq, there are NextSeq 550, NovoSeq. These are 2 like major instruments that they have, but they have also a list of MiniSeq, MySeq, etc., other sequences also. So long reads and short reads together will be used, and before for the hybrid assembly, and before going for the assembly you have to clean up these reads that we will see in this lecture, and also get an idea of the heterozygosity and polyploidy by using the raw reads, raw filtered reads. Okay, so basically another thing that we discussed in the previous class, the amount of sequencing that it needed for the short read based assembly and the long read based assembly varies and depends upon the genome, right? So short read you will be probably using a De Bruijn graph based method that we will be seeing in the detail in the future class, right? But basically this method depends upon a graphical basically connectivity between the K-mers that derives from the sequences, right? And that is why high depth will allow for higher level of assembly, better assembly. And similarly in case of long read, it is basically overlap, layout and consensus method, OLC, where the reads overlap will be calculated and which will be used to build the consensus. So basically these 2 assembly methods and many more we will be discussing in the future class. So I am not touching these in detail today, and we will be moving forward to the cleaning up of the sequences. So okay, so basically now we are clear with the long read, short read importance as well as their limitations. Okay, long reads are more accurate but may not provide a very highly contiguous or longer contig length, but and covering the genome, whereas long reads will be helpful in covering the genome as well as the may have more errors, but then short reads can be used to polish and correct those long reads. Okay, then paired end reads and the insert size between the paired end read is also important. Like okay, so generally 450, 350 to 550 base pair is the default insert size, but you can also go for 1000, 2000 more longer insert size, because the gap between the 2 reads. So basically we can generate only 150 base pair. Okay, so one DNA fragment I can read from both sides, forward and reverse, 150 base pair. So this is the area which I will not be able to read, because the technology only allows for this DNA fragment only 150 from this side and 150 from this side, forward and reverse, right? So but at least you will know that these are paired end reads which are coming from a single fragment. Okay, so in the future I will always know that the distance between these 2 reads, so this part and this part, okay, this distance I know because of the insert size, and then I will know that basically these two reads must go into the same fragment, right? So it will be highly useful when you are building a contig, right? And then you can fill up the intermediate, or basically this part, using other reads or other paired end reads, right? So paired end reads, that is why are very important, okay? And okay, so basically they are like commonly used, that is why in the genomics. Okay, so basically paired end reads can have multiple uses. One is to help you in the assembly. Second, like sometimes if you choose a very small insert size, this is your total DNA, okay, a fragment, you have done smaller fragments. So if I read 150 from this side, okay, and 150 from this side, there will be some overlap, okay, which can help you to assemble these paired end reads together also, okay. So which can produce, like if this was 150 base pair and this was also 150 base pair, so basically you can achieve like almost 250 base pair, okay, considering a 50 base pair overlap here fragment. And similarly, like so this way you can get longer fragments from a paired end read, which can be used for further building contigs and assembly, okay. And also like if you choose a even smaller insert size, and like for example in case of amplicons, okay, V3 or V4 or V5 regions, okay. So basically it is situation is like this, that you are reading from this side and also from this side, okay, the fragment forward and reverse, this fragment. So basically almost this fragment of DNA you have read in both directions, forward and reverse, and basically even if there was some error, it is corrected because you are reading in both the directions. So when insert size matters when you are using paired end reads for the genomic assembly, genomic sequencing and assembly. So moving towards the quality filtering of reads, it is very important because you have to remove the adapters, barcodes, primers, etc., which are associated with the reads, because during the library preparation you have to add adapters or barcodes based barcode for multiplexing, because you are mixing multiple samples together in a big sequencer, right, which can generate like several billion base pairs of data, right. And adapters because you have to bind those adapters to the flow cell, okay, right. So and you have to remove these because these are not part of your DNA or genome. And also like you also remove the low quality bases, because it is not that all these sequence fragments will be of equal quality. So there are quality matrices like Fred score, and based on that you can remove the fragments which are low scoring. Even from a read, single read also, there could be some parts, maybe start or end, which could be like having a very poor quality from at both the ends, and you would like to trim those sequences before proceeding for sequencing for the assembly. Okay, so like in this picture you can see like Illumina, so they are using adapters, okay. This is one adapter from both sides of the read, right. This is your actual piece of DNA that was inserted, DNA insert, okay. And there is also a primer which is used for amplifying or making the bridge amplification that you do in the Illumina technology, okay. So you have to remove these, okay, but before removing you have to find these in your raw sequences. That can be done by the FastQC, which is a software. So the good thing is that for all the genomic steps, genome analysis steps, if you know beforehand, there will be tools available for doing each and every step, okay. So you can basically construct a pipeline of your own by using a set of tools, like I will be telling many today and in the future classes. So those tools can be, if you know the order in which they will be used for the genome analysis and assembly and analysis, so you can use those tools in that order, okay, for any genome project, or for partial analysis by downloading data from somewhere else, right. And so basically using online may not be a good idea here, because you will be dealing with a lot of data which the online or the web based tool of the same thing may not cover. So downloading and installing in a local system, so having a good system, computational system is required here. Okay, so FastQC is one tool. So we have already done in the case of Linux that how do you install tools and unzip, install and put that in a path, and that you can also get this instruction like in case of FastQC or Trimmatic that we will see later. So I'm not going into details of how you will install these and okay. So basically there will be readme files, install files which can help you to install, and the same thing that we discussed in the Linux classes will apply here, and most of the tools have like same way of installation and okay processing, and you can get more information like by -- help option. Okay, so FastQC is one of the tools that will help you to do quick quality check on the raw sequences that you get from the sequencer. Okay, so basically from the sequencer you will be, after converting the sequencing into readable format, that is the FastQ format. Okay, in case of Illumina, right, you will be you can use those sequences with FastQC, which will give you a quick quality check. FastQC, so FASTA format and QC quality check, okay, of those sequences. Okay, and it will tell you what is the GC content of your sequence, whether it has adapter or not, and how many sequences adapters, right, and also detects the overrepresented sequences which may result because of the PCR duplication, right, because many times it could be that you may not be having sufficient DNA to proceed for sequencing. So you may like to amplify your DNA using the whole genome amplification or isothermal WGA, right, so that you can make get sufficient quantity of DNA. But in that process definitely you will be creating some bias by PCR, and also you will be missing out on some portions of the genome. But like if you really have limited DNA, then you cannot do much. Okay, so this is the FastQC tool. It can be downloaded from this site. Okay, and basically all the instructions to download will be available on once you download the package and unzip and untar it, and then basically there will be files, readme and install file, which will help you to run these commands. But the basic command that we will be running is like FastQC tool, then the output file name. Okay, basically I am also giving the complete path here. Okay, and then the list of files star.fq. So basically FASTA format, so basic FastQ format which you get from Illumina. Okay, and it can star means that all the files in that directory will be used together. Okay, so you don't have to specify each and every file, you can just say star.fq. so all the files in that folder with the suffix star.fq will be processed by FastQC. Okay, then Trimmatic is a software for the quality filtering. Okay, so because now you have estimated the adapters and okay, and the presence of primers and these in your sequences, then you will be now it is a task to clean or basically remove those unwanted sequence. Okay, even the low quality sequences, right. So Trimmatic is a standard software that is used for quality filtering of your data. Okay, it can crop the FastQ format of the sequences, maybe in FastQ format that you get from Illumina. Okay, and depending upon the librarian kit, there will be adapters and primers. So that information the service provider or the sequencer, your sequencing team, or if you have outsourced, then that company can provide that what barcodes or primers may be there. But if they have not provided, basically FastQC can tell, and there is a database of adapters and primers which are available. So basically these programs will be comparing against those databases, and mostly you will find your adapter and primer in that database. But otherwise, if that is not available, it will mark that as unknown adapter, or like that. Okay, so basically it is a very good software for read trimming and filtering. Okay, and the DNA sequences that you get from the sequencer have the quality values. Okay, for example, Fred score 10, okay, will refer to like every 10 base there will be one incorrect base call. Okay, that is very low quality, that means 90% accuracy you will get. And Fred 30 that we use, okay, that 1 in 1000, so one base pair in the 1000 base pair will be erroneous, and it will be referring to 99.9% quality, and generally we use this much strict threshold. Okay, you can go for like higher Phred scores also, okay, but basically this is one of the standard Phred score 30 we use for, because otherwise you will be losing a lot of data. But if you really know that your data quality is very good, and even at this level you are not losing much of the data, then you can go for higher Phred score also. Okay, so how to calculate this Phred score? Basically the program will give you an output that okay, these sequences that you are dealing with have this much Phred score, and then you can specify cutoffs that I want to keep only those sequences above this Phred score, okay, and you will be keeping those. So this is the tool Trimmatic. Okay, so it's a very nice trimmer for the Illumina sequence data. Okay, and basically Linux is the platform for all these tools because okay, even if Windows versions are available, it will not be able to use the power of your system efficiently. So Linux is the method of choice. So basically the Trimmatic will cut adapters from, okay, these are the various commands or basically information or parameter that you can give. Okay, sliding window of say 15, okay, basically it will be looking at your sequence in the sliding window of 15, okay, then another 15, then another 15, like that, okay, 15 base pair, okay, then maximum info like what is the read length and error rate, that ratio, okay, then whether you want to cut from the start or from the end, okay, based on the quality value threshold quality that you will define. That it could be that in the start the quality values may be very low, okay, so basically you would like to remove sequence from the start, and sometimes you know that okay, 5 or 10 base pair ups at the start or end I should remove like from all the sequences because they refer to poor quality, or basically to make the sequences of equal length or some based on your requirement. So you can cut those from start or end, okay. Crop you will use when you want to cut from the start, okay, and tail you will use when head crop will cut from the start. If this is a read, okay, you want to cut few base pairs from the start, you will use head crop. If you want to cut from the tail, you will use the crop, okay, and based on the like drop a read if it is below a particular quality, okay, so it will calculate the average quality and it will drop the read if it is below that, okay, and the Phred scores that you want to refer to are here, can also be specified. So basically Trimmatic also do one more thing, because you are using paired end reads, right? So it is not that after doing the quality filtering all the reads that you get or the data that you get, the sequence that you get will be paired end. So in some cases both the pairs, and basically in most cases both the pairs will be retained, because both the forward and reverse read, right, from the sequence, because it was paired end, both reads were above the quality score and all the parameters that you gave. Otherwise some reads, because this read was not of like the reverse read was not of the same good quality, so it will be dropped and only you will be remaining with this read, and sometimes this is retained and this one is dropped, okay. So basically that way you will have 4 type of files from the forward and reverse read that you have taken, they go to Trimmatic, and then basically you will get one pack or basically 2 set of files which will be forward paired and reverse paired, okay. That means that have the paired end reads above the quality score where both the forward and reverse partner are retained, or forward unpaired the reverse is dropped and reverse unpaired the forward is dropped because of the quality filtering. So basically this is the command to use it. So you in this command there are too many things, but you are specifying both the forward and reverse reads, okay, the folders in which you will be storing the data, okay. So basically like this is the file fast1 and fastq, forward and reverse. So 1 refers to forward and 2 refers to reverse, okay, and then output forward paired, okay, output forward unpaired, okay, output reverse paired, and output reverse unpaired are the 4 files that we have talked earlier, that basically 4 type of set of files will be retained. Okay, and then the clipping that you have done, or the trimming that you have done based on basically leading 15 base pairs, trailing 15 base pairs. So from the start and end, sliding window 15 that I have used, 15 15, that means okay, and how much I want to slide, when I what window I should consider. 15 is kind of default, otherwise you can also increase or decrease it. And minimum length 60, that means the read should be of minimum length 60 after you are done or with the trimming. Okay, so at least and all those reads which are above 60, okay, will be retained, and below 60 will be left out. Okay. So but basically you should also note that initial length that you start with the read was 150 in paired end, for example, but after doing all the trimming and all the quality filtering, so you are left with 60 base pair, which is also okay, because you don't want to lose a read, because that is coming from a genomic region, and if you discard too many reads, you will miss out on the genomic regions. Okay, so it can be downloaded from the web using this site. Okay, and it is a very nice tool for the quality filtering of the sequencing of the sequences, raw sequences. So that was for the Illumina sequences, but for the Oxford Nanopore data, another tool is called PoreChop, okay, which trims the adapters from both the ends, right? And I also looked at the adapters in between, because in case of Nanopore, there could be adapters may be at the ends, but adapter sometimes may also be in between. So it will split the read in between and divide that into 2 reads. And these cases you may also decide to either reject those reads which have adapters in between, but that way you will be losing a lot of data, because each read here is a few kbs or like even hundreds of kbs. Okay, so you do not want to lose those reads, even though they are chimeric. Okay, so basically if I cut them into 2 pieces, you may get rid of the chimeric, because it could be because 2 reads got joined together when they were passing through the pore, right? So there are several options like this. So PoreChop is available, published in Bioinformatics Advances, and this is a tool that you can download and use. Okay, download from this site, and basically this is the same that we have done in the Linux, the installation steps. Okay, so this information you will also get from the readme file. Okay, it's a Python based program, so Python 3 setpy install file, and basically PoreChop -help will display you all the commands. So you can match the adapters, trim the adapters from both the ends of the reads, right? Both the ends, forward and reverse. Okay, this is adapter, this is adapter, right? And split the reads with internal adapters. If the adapter is in between, you can split the read into 2, or discard the reads with having internal adapters. Okay, so various things you can do. So very simple usage. So PoreChop -i input file, output file name, -o output file name. Okay, there are spaces in between. Always mind that all the commands are separated by spaces. And then verbose 2, that means verbosity means how much information you want to display on screen when the program is running. So if you put it 0, it will not display anything, but if you put it 2, for example, it is here, means lots. That means each and everything that you will be doing, or almost everything important thing will be displayed on the screen, so that you know the progress, as well as that the program is running fine, right? Okay, and the number of threads. Okay, basically your system may have a lot of cores. Okay, so you can specify the number of cores. So the rule for number of cores is that, for example, you are using a 64 core machine, so you should keep some cores for internal processes. So you can minimally go for like maximum, you can go for like 60 cores, so that you can give 4 cores for doing the basic backend processes that the system needs, and 60 cores are devoted for this job. So if you specify like -t60, it will be distributing the process across the 60 cores. Okay, and it will become 60 times faster. Okay, so this is the case where there are reads. You can see this is highlighted in red. Okay, containing adapters. Okay, and along with the adapter, it will also chop 2 base pair which are attached to the adapter, just to make sure that adapter is cleanly got rid of. Okay, Okay, in case of link reads, basically proc 10xg, okay, so this is the tool, and it is also a set of Python scripts to clean the data generated by 10x genomic systems. So 10x is basically generating the link reads which are sequenced by the Illumina. Okay, so it can basically take the Illumina BCL FastQ files or even the FastQ files, right? And there are two steps, process and filter. In the first step, it will determine that what are the things to be removed, and the second step will actually remove those things. Okay, so basically again it is a simple way of running. So basically your directory in which the tool is there, the tool name proc process -10x reads.py, it's a Python script. Okay, -a, and then basically -1 is the input file 1, -2 is the input file 2. So reverse forward R1 and R2, forward and reverse reads. Okay, and the -o output file name. Okay, so basically it will look for the barcodes, presence of barcodes. Okay, first 16 base pair of read 1 and okay, and from both the sequence and basically for the quality values. Okay, and then it will label them. So it already has a list of barcodes, okay, or you can also specify or add to that list. So it will match the barcodes there. Match, mismatch, ambiguous means the barcode is slightly matching but still it is not exactly identical, and unknown, that means it thinks that it's a barcode but it is not sure, unknown. Okay, so it will annotate this file for each read and put that in the output, and then the tool that process this filter 10xpy will actually do the processing. Okay, where the initial file that we processed earlier, okay, okay, so will be used, and in this one you can specify that, okay, for example, at least minimum 100 reads should be there. You can specify like a lot more, but like you can say that, okay, if you think that something is a barcode, okay, if it is ambiguous or unknown, then it should be present in at least 100 reads. If it is not present in 100 reads, okay, that means it may be a random sequence or it may not be a true barcode, because 100 is like too minimal to be present in at least 100 reads is basically nothing, but it gives you an idea that, okay, like some common sequence is present in front of the read or back of the read which could be a possible barcode. So you may remove those. Okay, so this is one tool which can clean up your file. Okay, so basically your processed barcode file and -1 read the forward and reverse reads and the output file. Okay, then in a genome there are several things which are like important. The complexity, which basically is related to its complexity, the ploidy, the amount of repeats, and the heterozygosity. Okay, so all three things we have to determine that how much heterozygous the genome is. So because at one point in a pair of chromosomes, basically it could be in that, like it could be having more than one haplotype and there could be allelic differences. Okay, both the alleles are different. Okay, and which could lead to issues with the while doing the assembly, because you are either dealing with the short reads and using a de Bruijn graph based approach. So there it will matter even more, highly heterozygous genome, even if like it doesn't matter in case of long reads much, but in shorter reads it will definitely matter, because it will interfere in that assembly process. The ploidy level, how many copies of the genome are present. There are whole genome duplications like in case of wheat, okay, or basically in plants. So the amount of ploidy you have to estimate, and this will also help you to guide like how much sequencing is needed. Okay, and this is also an important task while you're dealing with the genomes, and the amount of repeats and what type of repeats and long repeats and short repeats. Okay, so that estimation also you have to do, but this part we have already covered in case of where we use repeat masker, tandem repeat finder, TRF, or emboss based programs. Okay, so determining the genome haploidy, polyploidy is very important, because it is first you have to report it. When you are doing a new genome, you have to report in your paper that what was the ploidy level. okay, and based on the ploidy and heterozygosity, you will be actually achieving some genome statistics. So a genome which is diploid and less heterozygosity, less than 1%, will be easier to assemble, but when these things increases, you also face difficulty in assembly. Okay, so basically in case of plants, this is a major problem. Okay, so a very good tool for this is Inquire, which is commonly used to estimate the ploidy. Okay, and it uses a Gaussian mixture model, GMM, okay, and calculates the maximum likelihood for the ploidy. So basically it uses three different models, diploid, triploid and tetraploid, and compare against all three, okay, and then tells that okay, which one could be based on the lowest log likelihood base, it selects that as the ploidy level. Okay, so basically once you have cleaned the sequence and you have got the trimmed clean sequence raw sequence file, so it will be doing the calculation using the GMM model, okay, and calculate the likelihood. Okay, so if you see here, for example, okay, so basically just read. So basically it compares that with the fixed model, and among the three ploidy levels, diploid, triploid and tetraploid, it says that which of these has the lowest log likelihood value. Okay, for example, in this you can see it has calculated the log likelihood value, okay, and you can see that tetraploidy showed the lowest log likelihood value, for example in case of the Jamun genome, which is recently released. Okay, so it will estimate the genome as a tetraploid. You can use like more than one method also to determine the ploidy level. Even experimental ploidy levels you can also check if they are already available in the plant C values databases, because basically you can do this both computationally and experimentally, and even computationally by multiple methods, so that you are sure that the ploidy level you are reporting is correct. Okay, another tool to estimate the ploidy is smudge-plot, okay, and GenomeScope too. So basically GenomeScope we will see in the later slide. So basically smudge-plot estimate the ploidy, and also using does the calculate the heterozygous K-mers. Okay, so basically this is a very good tool, which uses K-mers to basically look at the average K-mer proportions. So there could be like coverage of K-mer by A and B, two K-mers are there. So it will calculate the relative coverage of K-mer A considering all the K-mers, and similarly for K-mer B, and that is how it will calculate the ploidy level. Okay, so for example in this picture you can see that there are several K-mer combinations like AA, B, A, B, A. Okay, you can see in this combination of this three type of K-mers, A and B. Okay, basically their coverage is almost 62% of the total K-mers that you have. Okay, and you can see that at 3n, that is ploidy level 3n. Okay, so it is giving a peak here and a peak here, which tells that okay, basically this is the K-mer that is covering most of the K-mers, this K-mer combination, and so basically this base and since they are covering most of the K-mers, like 62% of the K-mers are representing the total coverage. So and at that level the ploidy level is 3n, so you can estimate that the ploidy level is 3n in this case, and basically this genome is estimated as triploid here. Okay, then there is a tool to basically count the K-mers, because you have to calculate the heterozygosity based on the K-mers. So there is one tool which is dedicated for counting the K-mer, that is the Jellyfish, which is available here and you can download from here. It takes the FASTA format file and calculate the K-mers in those files. Okay, so for after you have downloaded and installed the program, you can select that okay, you want to calculate the K-mer in both these strands, because the read can be read in both forward and reverse strand. forward and reverse complement of that. Okay, and then what should be the length of K-mer you can define, or you can also run it iteratively for several K-mers. Okay, then what is the memory allocation you want to give, because if you have say higher RAM system like 64 GB or 128 GB RAM, so it will you have to specify that memory size here. And basically this is also dependent upon the genome size. So if you are dealing with the genome of say 1 GB, so this allocation can be 1 GB or 2 GB you can keep for even like giving some buffer. Okay, then how many threads you want to run, 10 number of threads that is available in your system, okay, minus some threads which is dedicated for basic processes. So if you are running a system with like 64 threads, 64 total cores, so you can use 60 of those for this purpose. And the input file star.fq, that means all the files that are with the extension star FastQ format file, and the output file. Okay, so and then it is basically storing that in this output file, which you will be using in another program, which will be basically drawing the histogram. So basically Jellyfish histo. Okay, and this is minus threads you want to specify, how many threads you want to use, this reads file and the histogram. Okay, so it will give you a histogram of the heterozygosity. And generally like in case of plant genomes, heterozygosity is more, and less than 1% is acceptable and does not cause much problems in the heterozygosity. Okay, basically Jellyfish will give you data, or basically input file that you can use for the heterozygosity calculation by other tools such as GenomeScope. Okay, so GenomeScope will take input from Jellyfish, okay, to estimate the heterozygosity and also the genome size. Okay, so both GenomeScope and Smudgeplot were published together in Nature Communications, and they are widely used for this purpose. Okay, so they will take the K-mer profile from Jellyfish, and using that as input, they will be using those K-mer profile to generate the statistics for the genome assembly, that is the heterozygosity and the basically the K-mer frequency. Okay, so they are important tools to estimate the genome complexity and how much heterozygous the genome is. So generally like homozygous genome will follow a simple Poisson profile, whereas if the heterozygosity is more, you will get like a bimodal or those kind of complicated profiles. Okay, so just look at so basically this is the tool, it can be downloaded from here. So in this or you can also run it online. So just upload your files here. Okay, and K-mer length that you have used for the running Jellyfish will be used here, or what you found best using Jellyfish is used here. You have to determine the ploidy level, and you can keep the other information as default. Okay, and then submit. So basically after using the K-mer profile generated from Jellyfish, which you have given as an input, so it will be calculating the heterozygosity of various alleles. For example, for AA it was 99.4, whereas for allele AB, which is heterozygous, it is 0.576. That means the heterozygosity can be estimated by as 0.5 or 0.576, right? Okay, and because you got two peaks here, okay, one at 25x, another at 50x coverage. Okay, so basically two peaks means that it is heterozygous, and basically you are getting a low heterozygosity in this case, which is 0.576. Okay, and if it was not heterozygous, it would have given you like this number A would have been almost 100% here. Okay, so basically another thing is that because you got like two peaks, basically bimodal, so that means you are dealing with a diploid genome. So it is also giving an estimate of the polyploid in the genome as well as the heterozygosity, both things together. Okay, and the low frequency K-mers may be removed in this case because they could be sequencing errors. So that you have to take care at the initial trimming and quality filtering steps. Okay. So now another plot I am showing where basically there were 3 alleles like AAA, AAB and ABC. So and AAB and ABC are 2 heterozygous alleles, and a combination of the two like 5 plus 0.935, so 5.15 plus 0.935 will give you an idea of the total heterozygosity, and there are 3 peaks here. So that means the genome is triploid, okay, and basically the heterozygosity is 5.15 plus 0.935, okay. So that means it's a highly heterozygous genome, more than 6% heterozygosity, and will definitely pose a problem in assembly because it is heterozygous as well as a polyploid triploid genome. Okay, so in this lecture we looked at the usage of long and short read data, how we can trim and clean that data which can be used for the genome assembly, okay, and genome assembly part we will be dealing with the next lectures. But in this case we prepared the data for the genome assembly by quality filtering and removing reads which are of low quality or having adapters or removing the adapters, and also estimated the ploidy level and the heterozygosity of the genome, which will help us during the assembly, and also the K-mer count that we have generated, which will help us during the assembly process. And also these statistics are reported along with the genome paper, because for any new genome you have to report whether it is how much heterozygosity it has and what is the level of ploidy it has. Okay, so this is good for today. Thank you.